so my lab and me in general, we are very interested in studying 3' UTR. It stands for 3' untranslated regions. And I want to define now, so you know from the rest of my talk, these sequences go from the stop codon of an open reading frame of a messenger RNA. This is Pol2 transcripts, right? And they go all the way to the poly A tail, okay? So what happened is these sequences were ignored for a long time. People thought they're not important because they are not part of the open reading frame, and mutations within this region don't really cause much of a trouble compared with what happened in the open reading frame, so were ignored for a long time. But turned out that these regions are targeted by a zoo of factors, including non-coding RNA, microRNA, you maybe have heard about this, proteins, that target elements within these sequences and regulate the transcript stability post-transcriptionally. So this is a formal regulation that occurs after the RNA is transcribed. Okay? So I need to do a step back and explain you a little bit how transcription termination occurs, or what we know how 3' end formation occurs. This is something that you may be surprised for many of you. We know very little how the polymerase II senses that reach at the end of a gene. So we know a lot about transcription initiation. You know about how promoter works, you know transcription factors, the role of chromatin on promoters. You know about elongation, you know splicing, capping, all these events that occurs. But we really don't understand how transcription ends. And this is key because where Pol2 decide to stop transcription determined where the 3' UTR ends. Okay? So what we know, this is a mature messenger RNA. We have a cleavage site here, and eventually the poly A tail is going to be attached here. So we have few elements. One is just before the end of the messenger RNA, and other elements are immediately after the cleavage site. So this one here, we call it poly A signal element pass site, is canonically 2 A's, 1 U, and 3 A's. Okay? Then you have another element immediately after this is a less characterized enrichment of G's and U's within the region immediately after the cleavage. Okay? So these two sequences are recognized by very large complex. One of these complexes called cleavage and stimulation specific factor that they recognize this sequence here, bind this sequence here. Another one is the cleavage stimulation factors that binds on the other side. Then these two complexes associate together, they twist the RNA. Okay? Then this twist allows CPSF to induce cleavage of messenger RNA and recruit the poly A polymerase that attaches the poly A tail of the messenger RNA. Eventually, this sequence is going to be the messenger RNA, and this other sequence is going to be degraded because it's not, well, of course, protected, it's not capped, there's no poly A tail, and so on. So I want to start immediately with two experiments that really mark the point and why 3' UTRs are important in regulating transcription. So these are experiments that were done in the lab when I was at New York University from a postdoc that was there. I was just joined the lab. And basically, those are GFP experiments. So the person, Sabilal, was taking a GFP and expressed the GFP with a tissue-specific promoter in whatever tissue you want in this worm. And all she was doing, she was replacing the 3' UTR, adding either the 3' UTR corresponding to the same gene that she was using as a promoter, or adding a non-specific 3' UTR, a UTR that you know doesn't do anything, let's say. When you take these two constructs and you put it into the worm, you'll see it that the worm expresses GFP all over the places, like in both tissue, in both worms, right? In specific stages. But then you see late in development, you see that at some point the same construct, the light disappeared, the GFP expression disappeared, compared to this case where the GFP still stay. Okay? So what I want to remark here is that the 3' UTR is the only thing that is different between this construct and this construct. So this construct here, the messenger RNA is there, the GFP as an RNA is there, it just doesn't get translated into protein. Okay? So Sabi looked at the sequence and she found that there were like some sequences within the 3' UTR of this gene here that could represent a possible binding site for a microRNA. So she deleted this sequence here. And all suddenly, I mean, this is the graph here showing the GFP expression. If you mutated the 3' UTR in L4, you rescue the same kind of fluorescence that you rescue with a mutant, with a UTR that doesn't do anything to the expression. Suggesting that in this case, the 3' UTR dominates over the promoter to regulate gene expression for this specific construct. So you can have as much as RNA you want in this cell, in this worm, but if the 3' UTR is subjected to a regulation, that RNA never becomes a protein, so you don't have expression to that gene. Okay, this is key for the talk. So let me explain you why we use the worm, and many of you have never really heard about it, and we are kind of pretty big community at this point, but everything started in the 1960s when Sidney Brenner, and together with a few researchers, decided that in order to speed up research, they should come up with a new model system that allowed to do research in an easier way, in a better way than that was at that time. Okay? So they decided to look into a multicellular organism that have a very short life cycle, that can be easily cultivated, that are very small. There are relatively very few cells. These worms are very small. Let me have a small video for you just to get used to how they look like. They're very tiny. You need to have a microscope to see it. So they're transparent. We grow them in agar plates, just regular agar plates we use for bacteria. What we do, we put like a little bit of bacteria, overnight bacteria on top, because the worms eat bacteria, just regular E. coli. So, and what they do, they just stay on the plate and they eat the bacteria. In this case, it doesn't want to go outside because, of course, it wants to stay where the food is. And it stays around here. And it's very easy to cultivate. It's hermaphrodite, so with one single worm, in a few days, you have the plate full of worms. And they're all clone of their self. Okay? So just to continue, this model system was very successful. Even if you maybe are not exposed to it, a lot of the research you work on every day or that you're aware of came up out of the work done in C. elegans. So in 1964, Robert Horvitz discovered apoptosis. This is something I haven't told you. We know exactly how many cells this organism has as an adult. He has 959 cells. And he noticed that under 36 cells at some point die. And he didn't know why at that time. It took few years to discover that eventually those cells go into programmed cell death, and they eventually die. In 1993, Victor Ambros and Gary Rafkin discovered microRNA, the first microRNA, Link4. And then in 2002, Sidney Brenner, together with Bob Horwitz and John Salton, got a Nobel Prize for the decision to use this model system to study questions in biology. This was the first multicellular organism fully sequenced, way before the Human Genome Project was completed. In 2006, Greg Mello and Andy Ufai got the Nobel Prize for the discovery of RNAi. And many of you know about this very bizarre phenomena that cells have to regulate gene expression. Marty Chalfie in 2008 used C. elegans to demonstrate that you can express a GFP protein within an organism and visualize structure in vivo. And this was something also that he uses he got a Nobel Prize out of it. So the worms is very quick in growing. It's a very fast life cycle. In a matter of days, you start from one worm and you have thousands of worms. They have a very well-defined cell lineage. I told you, we know not only how many cells this worm has as an adult, but we know exactly from where these cells come from. We have all the lineage. So it started from 2 cells, and then it's going to divide. This cell's going to be this, and that, and so on. So you know exactly every cell of the worm where it comes from, which is, to me, is mind-blowing. We know that the worm is also hermaphrodite, so it's very easy to do genetics. The worm produces its own sperm cells and oocytes, so it's self-fertilized, so they're all like clone of their cells. It's transparent, so it's very easy to do genetics and look at the animals and study development and study the organogenesis, because you don't have to really cut the worm to see it inside. Again, a very well annotated genome. And what really we're interested in is that the UTRs in these animals are really short, so it's easy to look at them. Okay, on average, we have 200 nucleotides length of this UTR. In human, this number is up to 5,000, so it's completely different numbers. What turns out very interesting is that many human genes has been used and been shown that can compensate human disease in worms. So if you take one gene involved in muscular dystrophy and you express it artificially in the worm, you can create paralysis and you can simulate similar diseases in worm. So it, of course, is different model. It's not human, but you can please.
How long is the full genome?
It's 100 million bases, and we have 20,000 genes, so approximately similar to human. But in human, you have 3.5 billion. So the genome is very compressed, and the genes are all attached to each other. It's very common to find open reading frames that touch each other, the genes that overlap, a lot of isoforms.
And what what are those else? Are those like age? Or the life
So the worms, thank you for reminding me this. So the worm goes through 4 larval stages. The worm mold like snakes from one stage to another. So you have fertilization, the worms, you have a little bit of development within the animal, then is released, you have a little bit of extracellular growth, and then the egg hatches, the worm goes between L1, L2, L3, L4, and then eventually become adult, start producing his own eggs and sperm cells and keep the experiments. Every researcher in the world used the same worm that was initially picked by, it is funny, yeah, by Sidney Brenner in his backyard called N2, his Bristol worm. And it's funny because it's completely inbreeded. The sequence is all the same all the time. We know we have a perfect sequence compared with other model systems where sometimes you find changes that somehow it's difficult to make sense of it. So we were interested, this is something I want to present you a few slides about what I did as a postdoctoral work, so to give you an idea how you can use the worm to answer questions that you cannot ask to higher organisms, right? So we were interested in looking at these 3' UTRs and find out, how they look like, these 3' UTRs. What can we learn out of these sequences in this organism? What is the extent of their heterogeneity? How they, again, what is the length where they stop, where they start? And also why this is important. What can we learn out of this? Are there any elements that are important for regulating the expression of these transcripts? Can we detect them in worms? How these different 3' UTR are subjected to regulation in development, for example. And in the same time, can we develop tools to study 3' UTR biology? What I want to remind you and keep in mind for the rest of the talk, this is a living organism. He has tissues, he has organs. It's completely different from maybe the science you're used to, where you work on plates with cells and would need to grow, okay? So this is an organism that go through all different development, and he has it's a lot more complex than what you can think of. So we were part of the ModEncode, which is a branch of the NIH from the Human Genome Project. This was like a project that they developed parallel to the ENCODE project to sort of like learn about elements within the genome of the worms and flies and sort of like apply the technology and what we learn out of these model systems to human to sort of like predict or detect microRNA targets and intron exon splice sites on promoter regions and so on. Okay, so we did we integrated multiple approach. I don't know why this move doesn't work anymore. Multiple approach. We performed 3' rays targeting every gene present in human in the transcriptome of this worm. And I have a few slides on this. So the idea is that we take we design primer, we order almost 20,000 primers at the end and perform 3' rays sequences to extract 3' UTR from every gene from the worm. We also develop a technology in collaboration with John Kim and Michigan University to capture 3' end of 3' UTR and sequence them with high throughput sequencing method. And we do this at different developmental stages to find out how this length of the UTR changes during development. And also reannotated sequences that were already deposited at the DNCBI and so on. So this is mostly a bioinformatic problem. You have tons of data from many different data sources and you want to somehow fuse them together and make sense out of your data, right? So it took really a lot of time to make sense of all this data. But we ended up producing a very, very well-defined 3' UTR-ROM of the worm. We detected almost 130,000 distinct poly A sites, so where the UTR ends. This is supported by over 3 million of independent tags, so it's very deep coverage. We detect 3' UTR at least for 85% of all experimentally supported protein-coded genes. And what is key is that we have the complete sequence from start to end of every UTR, so we can look at things like RNA editing, splicing, and so on, right? And doing this, we ended up revising 40% of the gene model in worms. So these are the kind of things that came up and were really interesting for us when we did the analysis. So if you remember, this is a cartoon I showed you at the beginning how 3' end processing occurs here at the cleavage site, and I mentioned that this element here is canonically AAUAA8. So it turns out when we do these experiments that in worms it's not always AAUAA, tend to change. You have only 40% of genes possess this element here. For the majority of them, you have either 1 or 2 permutation of this canonical sequence. Okay, this was something bizarre. We didn't understand why at that time this occurs. And we noticed there is a large population of sequences that we couldn't detect any pass element. Okay, so the way we did this analysis very blindly, we took 50 nucleotides from the stop of every UTR that we cloned and we sequenced, and we looked for enrichment of hexamers, random hexamers. So this is made by computer, purely by computer, and that's how we built this kind of structure. Well, what turns out very interesting is that the cleavage is very precise. So this pass site is exactly a -19 of the end of every 3' UTR. This is something turns out to be also true in human and in many other organisms. And here we plot the canonical site AA, UAAA, that is here, together with all the variant paths that we detected in the sequences here. Something that really came up and out of the blue, and we were not expecting it, is that it turns out that and this is something will come back more and more during the talk, that open reading frame come with many different 3' UTR isoforms. This is something that we didn't really understand at the beginning. So what happened is that the same messenger RNA can possess multiple 3' UTR isoforms. Okay, so it turns out that nearly half of the genes in C. elegans use these multiple polyase signals. Almost 61% have only one isoform, but sometimes you have 2 isoforms, 3 isoforms, even up to 5 different 3' UTR isoforms. And this is something that turns out to be also true in human, but let me drive you slowly on that. So this is what I mean with multiple alternative polyadenylation. So you have cases where you have multiple elements within the same 3' UTR, and in some tissues or in some developmental stages, sometimes the complex CPSF recognize this element here and cleave the UTR here. Sometimes recognize this long UTR, this element on the other side, and cleave the UTR there. So you ended up with 2 different isoforms in this case. Okay, this is happen most of the time, 65% of the time we have this activity here. But you can also have cases where you have alternative CDSN, so you have different splice events that occurs in the open reading frame that create frame shifts, and you ended up with different 3' UTR because the stop codon now is in different position. We have something very interesting. We're very surprised about this. We have splicing within 3' UTRs, and we don't really understand why you want to splice out an element out of a region that is not translated into protein. We studied this very carefully, and we looked over a small group of these for 79 examples, okay? And these are cases where you have intron retention. So sometimes the intron is spliced out, like in this case here, sometimes it's kept, okay? Maybe in a different larval stage, different developmental stage, and so on. So we looked a little bit carefully on this. This is an example. We give you 2 examples here for 2 genes, PAR5 and SAMS1. In this case, you have the 3 different exons that are here, and sometimes the one spliced is more abundant, sometimes the contrary, the one not spliced is more abundant. So we looked at this case, and it turns out that the majority of them, they have elements within this region that is spliced out, that is not present here, but it's present here, that suggest a possible binding element for regulatory complexes that can load on the RNA and repress the translation of the RNA. Okay, and this is a concept that's going to come up in few slides, and it's going to be clear for you in few slides. So we didn't really understand why you want to have multiple 3' UTR isoforms and how the machinery that cleave 3' UTR can actually decide which UTR to cleave. No, you want to have a short or the long, right? So when we look at UTR that genes that contain only one 3' UTR, we notice that the canonical element AAUAAA is present for almost 60%. The majority of the UTR that are present as a single 3' UTR isoform, you have only one element here. But then, if you have cases when you have 2 different isoforms, in this case a short form and a long form, right? The long form always possesses a canonical form. It is enriched for canonical. It's very similar to what you get when you have a single one. But then, the 3' UTR, the short 3' UTR isoform possess different element. So the canonical here drop dramatically, and you have an increase in the other 2, so which are the variant paths or the no paths I was mentioning to you, suggesting that maybe this is how this complex, the complex that cleave 3' UTR and release messenger RNA discriminates between the 2 different UTRs. So you have a case where very abundant, very ubiquitously expressed element, and the complex that cleave these is maybe present, mostly present in every cell, in every subtype. cell subtype, so that's why you have it here. Of course, you don't want to have it here, because if you have it here, you will never end up with a long 3' UTR. So you keep maybe accessory factors that recognize these other elements, these variant paths or these null paths that eventually cleave and reduce and shrink the 3' UTR. Okay?
I have a question.
Please.
So the ones that had multiple isoforms,
Yeah.
did you find multiple canonical elements with those?
Yes.
Okay.
And we don't really, I'm going to present a case in few slides of that. Okay. Yeah. We suspect that most of this regulation occurs at tissue-specific level. And this is what I'm doing now currently in my lab. We're trying to deconvolve all these big 3' UTR library in small groups and find out where each single UTR is expressed, and eventually study the regulation that the UTR is subject. Because what we think, and I want to anticipate for you now, is that the reason why you have these two isoforms, short and long, while the operating frame is the same, is that you may have elements here that maybe are important for regulation of the messenger RNA. And when you want to escape this regulation, you just make a short form so you can escape this regulation here. We have few evidences for that. Okay? So in the meantime, other people worked on this. This is a lot of work. A lot of people work on trying to understand how the 3' UTR in worms work. So they validated, this is Bartel group in MIT, so he looked on validating 15,000 of our 3' UTR isoform. He detected 9 more thousand 3' UTR isoforms, so there's really a lot more that we haven't really highlighted at this point. Now, I want to switch a little bit on what we're doing now here at Arizona State, so you have a better idea where we're going and the observation. So Steve is a graduate student of first year. He just joined the lab, and he's very interested in studying this mechanism of alternative polydelineation and 3' formation. And together with a very bright undergrads, they are in the lab and they're doing experiments trying to address these questions. So alternative polydelineation turns out to be really widespread. It's not present only in worm. We are highlighted in worm together with other groups, but turns out that every organism you can think of has multiple 3' UTR elements. Human, mouse, dog, and plants, and even Cerevisiae, if you think about it, a single cell organism still have multiple 3' UTR, which we really is very surprising to us. So this is the question that Steve wants to answer. Look at this case. This is a gene, it's a MYO2, it's a myosin gene. Okay, it's mostly expressed in the pharynx of the worm. For this gene here, this is the end, this is the stop codon. We detect 1, 2, 3, 4, 5 different 3' UTR isoforms. Okay, in the specific case, the long one uses a canonical AAT-AAA element that maybe is present, that is present within this region here. But also you have another element repeated here and the same element repeated here. So you have the same AAT-AAA repeated 3 times within the same 3' UTRs. But somehow you detect the short, sometimes the medium, sometimes the long. Okay? So what is interesting in understanding is that why you need to do this, why you have multiple 3' UTR that are really precisely cleaved. It's not something that it's cleaved just because you need to release the messenger RNA. There is a reason why the cell cleaved there. We just don't understand it. We want to study how, not just the function, but also the molecular component that actually allow you to do this. And in the same time study how this alternative polydelineation is regulated in the cell. Okay? So in order to do this, what he wants to do is to look into tissue-specific RNA libraries from various tissues of the worm. So when we did this and we highlight all these 3' UTRs, we did as a whole. We took the worm, we lysate, extract total RNA, and we sequence it. We did this in different larval stages because it's easy. You synchronize the worms, you release the synchronization, you collect different time point, you release the RNA, and you sequence it. But we still detect the same 3' UTR isoforms in the same, different 3' UTR isoform in the same pool. So we suspect it's something to do at tissue level. Now the problem here that this is a living organism that is very small, very hard to work with. You can't really go there with the blade and cut it and recover organs and sequence them. Okay, I want to appreciate how hard it is to do this kind of experiments in worm. And again, you want to identify these 3' UTR isoforms and eventually study their behavior in development and what kind of factor regulate one 3' UTR versus another, and at the same time regulate gene expression in the worm. Okay? So in order to do this, we came up with this idea. We took advantage of the fact that there is a protein called polyadenylation binding protein that normally binds poly-A tails of mature messenger RNA. This is an example what happened during a translation. You have a mature messenger RNA that is slotted on the ribosome. This is the cap, this is the poly-A tail. This protein normally helps the circularization of RNA that is normally important during translation, allelic translation. So this protein binds the poly-A tail. So what we did is came up with this strategy. We take GFP and we fuse this with the polyadenylation binding protein of worm, PAB1, and we fuse this to a 3X flag, and we attach a UTR that we know is not subjected to any regulation. Okay? We take this construct and we express in a different tissue using a different tissue-specific promoters. Sometimes we express in the intestine, sometimes we express in muscle and in other tissue. Right? The idea is that when you express this construct, let's say in muscle, in the pharynx, like in this case here, the RNA present in this specific tissue possess polyadenylation binding protein which is flagged. Okay? So when you take the worm and you lysate the worm, you can do an IP for flag and recover, really fish out the RNA present only in this specific tissue and eliminating the RNA that is present in other tissue. Make sense? So that's what we decided to do, to basically identify tissue-specific promoters for genes that are expressed specifically in one tissue and confer identity to that tissue. We decided with epidermal, digestive, reproductive, muscular, excretory, and nervous system. And we ended up producing 8 lines. Now I'm going to give you a few examples here. One we started is JES1. It is the promoter of JES1, so express the construct in the intestine here. As you can see here, there are like 20 cells. So we're talking about very limited number of cells here. So you also need to appreciate these. We express this, and the good thing is that previously researchers used a sequence the population of RNA present in the intestine, so this also provides us a nice control for the experiments. Then we express it in the muscle using MyoTwo. Again, the muscle is one of the brightest and the easiest organ to detect, the pharynx. This is the mouth of the worm, the one it uses to intake the bacteria out of it. And then we try to push this as much as we can. In this case, we looked on excitatory neurons. These are really 3 neurons present in per worm. And we want to try to see if we can actually fish out RNA only from 3 neurons out of a single worm. And you have cases like a GABAergic interneurons where you have around 20 neurons, or you can see them all lined up here, and so on. So the idea here is that we take the construct, we inject in the worm, we force the worm to integrate this DNA within their own germline, so they start producing worms expressing this construct. Then we grow liters and liters of this worm, really liters. And then you recover the worm, you spin them down, you lysate them, you do the IP, and you recover the RNA. So, and then eventually you sequence, you do IEEE sequencing, and you discover these UTRs. This is what we are now. So unfortunately, I cannot give you finalized numbers. And we are now trying to find, to decide, to look at the specificity and the sensitivity of the approach and optimize that.
It looked like, sorry,
Please.
It looked like on your gels that the length for each tissue was relatively the same, correct? It was like 1,500 bases.
Say it again?
If you go back, so the gels on the side there.
Oh, this? Yeah, the cost is the same.
Yeah, it looks like they're the same for all the tissues there.
Yeah, the cost is the same. All we do, we just use a different promoter.
Okay.
So I show it to you. Sorry, maybe let me just explain it here. So this is just the PCR. So the way it works, if you inject DNA in a worm, the worm this is a living organ. It's not a cell that starts dividing and producing a population of cells. So this one here is going to stay in that tissue. It's going to stay till the worm dies. So you need to somehow make sure that this piece of DNA is incorporated physically in the genome, right? So in order to do this, we use a very complex mechanism that basically is based on a transposon the worm has normally. And basically, we have this transgenic line that possesses this element within an intergenic region of chromosome 2. So you take your construct, you inject physically into the gonads, hoping that this construct get into the germ line. So eventually, that single cell is going to have the recombination event, of course, and eventually all the worm from there on is going to be transgenics with that gene. So it's a very tedious and long process, take months to make one line. Then we do a PCR with primers within this intergenic region to make sure that the amplicon makes sense, and basically that's why the band is always the same at the end of the day. And then we do an IP with the flag type, but the construct is always the same. All you change is the promoter, so where you express it. So every construct, we did the Western only on the first, I think, because then is the same thing all the time. We just test the presence of it. So the open question is okay, let's say we can identify where a specific 3' UTR is expressed in which tissue. How can we learn what's the role, what's the function of this UTR in the organism? What kind of elements are important for the regulation of this messenger RNA? Okay? And then I'm going to introduce you a little bit of microRNA. I don't know much this audience know about microRNA, but pretty well characterized nowadays. There's a lot of interest in microRNA, also in cancer research, not just in model systems and so on. Those are small non-coding RNA that target specifically the 3' UTR of mature messenger RNA and target messenger RNA and at the same time repress translation of the messenger RNA. Okay? They are important. They've been discovered in worms, and they're important in regulating for development and morphogenesis. So they're very short, almost more than 20 nucleotides, 22 nucleotides long. Okay, this is the beauty of the worm, because this thing has been discovered in worm. This is the level of RNA and protein of specific proteins, Lin14, Lin28, and so on. And this is the larval stage where you detect the specific protein and the specific RNA. So at larval stage L1, you have a lot of proteins for these two genes, Lin14 and Lin28, right? Then at the transition between larval stage and larval stage 2, what happens, you have one of these microRNA, Lin4, that start expressing it, start being present. And when this small RNA comes up, this small RNA targets specifically the 3' UTR of these two genes, Lin14 and Lin28, and repress the expression of this gene here. In fact, the gene now, the proteins from Lin14 and Lin28 drop dramatically, okay? And this step is key. If you don't have if the microRNA expression doesn't rise, the worm doesn't switch from L1, L2, okay? This is very important. Think about it. A small piece of RNA, 22 nucleotides. See how much important it is for worm development. Okay, the worm doesn't go from L1 and L2. Then what happens is that this microRNA Lin4 makes sure that not just these genes stay down, but also maintain the presence of other genes, including like LED7, for example. It's another microRNA that comes up here. The point is, this microRNA target the 3' UTR and repressing in different stages, but it's not specific one microRNA per 3' UTR. Sometimes the same microRNA can target multiple genes, in this case Lin14, Lin28, and also Lin41 here later. So you have two microRNA, Lin4 and LED7, that come up at different stage points, one in L1 and one in L3, late L4, that control when the worm has to mold from one stage to another, okay? Now, microRNA, the beauty of microRNA, that those are genomically encoded. Human have approximately 1,000 of these sequences. Worm has around a few hundreds, no more than that. They are originated by Pol2 transcribed, so they have either the cap and the poly A tail. They initiate as a precursor, which is a hairpin that eventually is exported in the cytoplasm, is recognized by dicer. Dicer cleave it, release the two strands, and use one strand and load one strand in the RISC complex, that is the complex that is involved in silencing specific messenger RNA. And the complex can either interfere with the translation, repressing translation, or induce the messenger RNA cleavage and basically take rid of the RNA. Okay? What is key here is that the way this microRNA target the 3' UTR is not 100% conserved. It's not really a perfect pair between the RNA and the 3' UTR. There's a little bit of imperfect match. Okay. Now, why this is important? It turns out that microRNA, it's not just important in worms, but turns out to be present everywhere in any system, in any organism that you can think of, but especially in human. It turns out to be really important in controlling key aspect also of cancer and in other diseases. This is a paper that came out in 2009 from Bertel group again, where they looked on the length of the 3' UTR in different cancer subtypes. And the idea here is a UTR tend to be really, really short in order to escape this regulation through microRNA. MicroRNA normally target is 3' UTR. Since the cells has to go into an active cell division, UTRs tend to be really, really short, so they can escape this regulation. Okay? Now, people looked at this microRNA. It turns out to be present in important many different cancers. Sometimes they overexpress, sometimes are downregulated. People talk about microRNA signatures. Sometimes people is able to identify primary tumor or secondary tumor based on the stage of the tumor based on the microRNA profile, the presence of this microRNA, and they are present in several cancers. One that we really like to use in the lab for our experiments, and I'm moving a little bit from worm to human now, is a miR10B, which is a microRNA to be found very, very highly expressed in aggressive breast cancer. And this gene here, this microRNA miR10B, seems to be important in the regulation from in the metastasis process, in inducing the metastasis. So, please.
Can you go back 2 slides?
Can you see from there?
I can't now.
Okay. This?
Yeah, here.
Yeah.
What were they doing with cyclin D1?
Okay, so this is an example. I mean, of course, he published the best example he had. He did a northern, and he uses 3 genes. In this case, a cyclin D1, IMP. I don't know what this gene was about. This is DICER1, and he looked on the first line you see here is the normal tissue, and then let's say in this case here you have breast, and then you have all different cancer cells, different breast cancer lines. It's all lines, okay. And the argument he wants to make here, and that sometimes the UTR shift in length, like in this case, okay, you have in cyclin D1, you have the same length here, the length disappeared, but then for these other genes, you have the contrary. For this gene here, let me see. For this case here, for DICER1, you see you start having isoforms that are really short, and you have a very long isoforms here on top. You start seeing shorter isoform here, also short isoform, increasing in short isoform compared with the normal tissue, this normal tissue, this normal cancer, this is cancer, and so on. So the idea, the argument he made is that UTR tend to be really, really short because they want to escape any form of regulatory regulation within these cell lines. So being short allows the cells to constantly produce more RNA and escape this regulation, so the cells can produce more RNA and more protein. He's still debating this in the field, but he proposed this a few years ago, and now it's it's accepted commonly that normally proliferated cells tend to have very, very short UTR, while cells that differentiated, they tend to be really, really long because they need to have this regulation through microRNA to fine-tune development.
And did these include the length of poly A tails as well?
In this experiment, they haven't done that, but there's a lot about poly A tail in worms. So of course it's regulated. The length of the poly A is regulated. We have poly A polymerases in the cytoplasm that control how long the poly A tail. And longer it is, the more active translation is. But I don't think he looked into the length of the poly A tail.
I asked because we just did this tumor microRNA experiment, and I used some prediction algorithms like target scan and pick target to predict of like the 10 microRNAs that were at the top of the list.
Right.
What genes do they target? And cyclin D1 was targeted by like 9 of them.
Right.
It was weird. And RB1 was targeted by the same microRNAs.
Mm-hmm.
Like
I mean, to me, those are very difficult experiment to interpret it anyway. You know, when you have this kind of, you always need to compare this with the proteomics and find out if you really the RNA repress the gene and the gene is not there as a protein anymore. Because if you do microarrays, you detect the RNA levels, but doesn't mean that the RNA is not downregulated in becoming a protein through microRNA or the regulation from a regulation. So they're very difficult experiment to detect at that point. But let me tell you more about this. So what people think in the field is that you may have multiple genes and you may have microRNA that maybe target one form and another form and another form, because the sparing is not 100% conserved. You can have these loops around. So because the nature of this microRNA, it's very difficult to predict where this microRNA binds within the 3' UTR. So if you're interested in studying 3' UTRs and you want to look for these elements, you need to come up with common rules that tell you where this UTR, where this microRNA or this RNA binding protein, which is another big story, target the genes. And the reason why I'm saying this, I'm going to go back a little bit on Lin4 and Lin14 sites. This is this is the open reading frame of Lin14. This is the UTR of Lin14, right, in the poly A tail. So Lin4, this microRNA, that the first microRNA discovered, this is the sequence of this microRNA, can recognize multiple elements within the same 3' UTR. Okay, so sometimes you can form very complex loops here because the pairing is not 100% conserved. So you can really target, the same sequence can target really hundreds, if not thousands, of different RNA. So what the holy grail in the field is to really start understanding what are the rules that control microRNA pairing with the 3' UTRs. Of course, I was mentioning to you, if you go to the hallmark of cancer, people found all sorts of microRNA that are either overexpressed or downregulated, that can act as a tumor suppressor gene or oncogene and so on. So there's a lot of interest in understanding, what are these microRNA controls, what kind of genes, what kind of class of genes this microRNA controls. If now I know it's overexpressed, I know that the pairing is not 100% conserved within the targets, can I somehow predict or study where this microRNA is supposed to target within the UTRs? So nowadays, the only thing you have to actually detect microRNA targets is using predictions, so computer software that allow you to predict how much you think that the microRNA target the 3' UTR. Okay, so the way they work is basically take advantage of what we call the seed region, which is the first 2 to 8 nucleotides of the microRNA when it pairs on the messenger RNA. So the seed sequence of the microRNA is supposed to be 100% conserved. You need to have 7 nucleotides that are 100% conserved to the target UTR. And we think the reason why you have these 7 conserved sequences is because physically when the Argonaute protein load the microRNA on the targets, use this to really dock on the messenger RNA. So the first nucleotide, the first 7 nucleotides will be 100% conserved. Then it's more let's say, different people use different rules to decide how much it thinks that microRNA targets are not the UTR, okay? Most of them are based on sequencing comparison. So they take multiple species, align multiple species, they look for block within the UTR that are highly conserved, they look for the seed region within this block, and then they know how many microRNA you have, so they start running prediction based on thermodynamic stabilities and so on, to sort of like predict how much they think this microRNA can be target, this UTR, and so on. So the most important, the most used one are Miranda, TargetScan, and PICTAR nowadays, and the most used. They are quick, inexpensive. You can go on a browser, you can just put your sequence of the UTR. It tells you what are the microRNA the most likely targeted UTR, and so on. The problem is that they don't really target. So they cannot detect non-conserved target. If the target is specific to human and is not present in mouse and other organisms, there's no way you can actually predict using it through this software. Okay. Also, they cannot look on non-canonical target. So we talk about the seed, how important it is. It turns out that a lot of microRNA now target regions that do not require a seed or a pairing, 100% pairing also through the seed. So those microRNA pairing, you lose it using this kind of software. Okay. Then turns out that a lot of microRNA now recognize sequences of RNA that are edited. So this, of course, you cannot predict because you can't really detect a seed or a sequence of RNA that changes. You don't know what sequence changes and so on. So the take-home message is that the majority of the target, of the microRNA target, are still not validated. So we did this exercise in the lab. We looked on some of the most common microRNA present in literatures here, in this case LET7C, miR10B, and so on. And we ran PICTAR, and we asked the software, tell me all the targets you think you have for LET7, and we plot them here based on the E-value that the software returned to you, right? And we draw here the mean. And then you see we did this for LET7, for miR10B, and so on. So in LET7, the software detects 620 targets. So those are 620 genes present in the human genome that the computer thinks LET7, this small microRNA target. Then when we actually went to the literature, we did a very in-depth research looking at all the papers that actually validate few of these targets, right? All the papers that people do wet bench approaches to actually look for these microRNA targets. And it turns out that there are very few, like in this case for LET7, you see you have 1, 2, 3, 4, 5, 6, 7, 8, and only 9 validated targets are actually predicted by the software. And also something that really surprised us, also the confidence score is really low. We were expecting that the one with the very confident, they would be the first to be discovered, or the first people start looking at, because maybe they have a clear importance in disease or in other things. But then, and this was surprising, we found a lot of papers, in this case for LED7, of genes that are known to be targeted by LED7, and they have actually experimentally validated the pairing, that were not predicted by this software. So LED7, for LED7C, PICTAR predict as a 53% false negative rate compared with the, those are targets that you will never detect using this software here. So we looked on target scan. Target scan is actually detect more. It does a better job. It predicts 35% of false negative rate. So you still have genes that are validated but not predicted by the software, very few. But you have to say it predicts 1,000 targets compared with the other to PICTAR that predicts only 600 and so on. So you have more predictions. So you have a low false rate negative, but you have an increase in false positive because you have more data. So this is just to give you an example. P53, a lot of people work on this gene, a tumor-suppressive gene mutated in most cancers and so on. This is the 3' UTR of P53, right? And those are all the conservation of 5 species in human. Of course, you have an enrichment in conservation within the polarization signal element. This is important for producing the cleavage, so you need to have this conservation here. But overall, the conservation is very low, and in the open reading frame, you have it. So target scan predict microRNA targets in this position, this position, and this position, and that's it. So we looked in the literature, really searching, mining every paper where a researcher tested and detected any microRNA that physically target the 3' UTR of P53. And turns out that you have 1, 2, 3, 4, 5 microRNA that are known to target P53, but cannot be predicted using this software, because they lie with regions that have very little conservation, and it's impossible to predict with this software because of that. Okay, this is the problem that we have. If we want to detect microRNA targets in a region in a sequence that is around 2,000-3,000 nucleotides long, and we want to detect a small piece of sequence that is 22 nucleotides long, how can we do that? Okay. So this is the let me just zoom this. So this is the questions that we have. So how can we detect microRNA target using an unbiased approach, right? How can we identify specific elements that are targeted by microRNA and really study what's the contribution in gene expression, and how can we then move this thing forward and looking on therapeutics and using a study and apply what we learn out of this pairing between microRNA and 3' UTR to disease, like for example cancer initiation and progression. So Justin is another graduate student. He's also first year graduate student. He's actually doing this project together with Alex, which is our tech in the lab. So they both took this very challenging project to sort of like develop a new high throughput method that is able to detect microRNA targets in vivo. We call it this project 3' Life, that stands for high throughput luminescence-based detection of functional elements within 3' UTRs. And I'm going to tell you a little bit out of this. So what we wanted to do is to develop a technology that allows you the rapid and systematic screen of functional microRNA targets in human 3' UTRs. Okay? So we want to develop a technology that precisely maps these 3' UTRs, and a method that is simple, that can be detect this element at single base resolution, and also can be eventually scaled to look also for sequences that are not just microRNA, but also RNA binding protein, which is another very big story that unfortunately I don't have time to talk about this now. So what we came up with, to sort of like go with a binary testing, 1 3' UTR with 1 microRNA. Test the interaction, detect this interaction using luminescence. If the interaction occurs, we save the gene and we retest in the second screen. If it doesn't work, we leave it and we go with the next one. And ideally we screen the whole human genome, and we clone every 3' UTR, of course, of the human genome. The idea is that you look on the 3' UTR, you design primary, you clone the UTR, you prepare a vector, basically using a dual luciferase reporter assay. At the same time, you have to clone the test microRNA that you want to test. You design primary, you amplify out of the genome, and then you move it in the vector, and then you pair 1 to 1 and test luminescence. So of course it's done in 96 well plates. Is everything automated? There at the biodesign we have all these toys to do all these kind of experiments. So we decided to start with a pool of 2,000 cloned 3' UTR from human. Those are the complete data sets of transcription factors 3' UTR in human. They are approximately 200, 300 transcription factors, depending on who you talk to. And the idea is that, 2,000, so it's like 20, 30 plates, and it's very easy to manage at the end of the day. And the idea is that you take a test, you put a control, you allow the pairing. If you detect interaction, you have a change in luminescence compared with the others, okay? Everything is automated, it's performed in 96 well plate, and this is how we ended up doing it. It took really quite a bit to optimize these vectors. So it's a dual luciferase assay, so you have a Renilla luciferase and a Firefly luciferase within the same vector, okay? You have two different promoters, in this case a CMV promoter, or PGK promoter here, and we put a 3' UTR, unspecific 3' UTR on the first color, and then we put a test 3' UTR inside in this position on the second color. So for this element here, MS2, I'm going to talk about a little bit next in a few slides. But the idea is that if you take this construct here, you put it into the cell, you transfect it in cells using HEC293 cells, the first color is going to give you the transfection efficiency. The level of transfection is constant all the time. But the second color, if you have a microRNA that targets the 3' UTR, is going to lower the expression of the second color. And then you basically study the balance between the two different color. It's going to give you a rough idea how much activity that specific microRNA has with this 3' UTR. Okay? For the second, for the microRNA, the probe microRNA that we use, we ended up using this very this construct here. Basically, we put DS red, and we put the microRNA within the 3' UTR of the DS red within 2 introns present on the border of the construct. The idea here is that you have a promoter that drives the color, the red, so it's easy to detect in the microscopes, but only the cells that are actually transfected, they're going to have the microRNA expressed into the cell. So you actually can use this to normalize the reaction after you do the transfection, because since you do it in high throughput, you can really control how much transfection you have in one well versus another. So the pipeline is design primers and amplify these primers and shuttle in the dual luciferase reporter assay. At the same time doing this for the pre-microRNA again, then perform the dual luciferase assay and test for positive screen, and eventually come up with a rule that tell you this specific microRNA recognize this 3' UTR, this other 3' UTR, and this other 3', and start building this network and find out, what microRNA control, which 3' UTR each microRNA controls, right? So we tested this with the classic interaction between Lin 28 and Lin 4 that just mentioned it to you. So this is the ratio, the numbers here just tell you that the normalization between the two colors. You have a Lin 28 alone. When you add the microRNA, the luminescence of the second color just go down, so you have significantly lowering expression. If you delete the microRNA target, you actually rescue that, and you have an increase in that. And if you delete the microRNA, you add the Lin 4, similar comparable data. So what we decided to do then, when we optimized that, we decided to focus on two different microRNA that have a cancer-related microRNA, LAT7C and miR10B, that were shown in many papers to be involved in many different aspects of breast cancer. So we amplify these sequences, we clone into these vectors, and then we are ready to do the experiment. So one of the problems we had is to how we clone this 3' UTR. We basically need to extract 3' UTR for 2,000 genes from human and clone it in an expression vector, in our expression vector. So the gene models in human is very, in my opinion, genes are not very well annotated compared with worms. And I say that mostly because in human you must have cell lines. So you have different cells, and maybe they are cancer cells. So you have splice isoforms, genes, truncated genes, and in the same time you have many different databases using different name for the same gene, or they have complete different nomenclature for the same gene. Then you have different versions from the same list of genes. So it turns out to be really, really complicated for us to do that. It took quite a bit. And one of the problems we had, we didn't know what the best approach was. Do we use 3' rays? or do we use cDNA libraries, or do we directly amplify from the genome, right? So the problem with 3' rays and cDNA libraries, the 3' rays problem is that which cell line you're going to use. You know, you're going to have maybe a cell line where you have a specific 3' UTR isoforms. The idea is to take the longest possible so you can actually do your experiment and look for these elements. So we decided not to go with the rays. cDNA libraries normally are enriched for a specific tissue. So again, we're going to have this bias and we don't want to go through that. So we decided to look into genomic PCRs. And this is how we ended up doing. So this is a gene. We designed two primers, one that lies just before the stop codon of every open reading frame here of these 2,000 genes. One, we decided to go under 50 bases after the longest 3' UTR isoforms detected for that gene. And the reason why we want to leave this extra space, because I told you at the beginning that messenger RNA has elements here that are important for the cleavage and polyadenylation of this UTR. So we want to make sure we include these elements so this UTR is correctly spliced and correctly produced.
So how are you detecting the end of the UTR?
So this, of course, relies on gene models. We ended up using RefSeq, which is not the best in my opinion, but is the one that make more sense. Because it's the one that make more sense at the end. The problem we had is that how you define the stop codon, because sometimes you have multiple isoforms within the same gene. So this is one of the major problems we have. So if you have this is computationally though, but if you have a stop codon, we take the isoforms that correspond to the stop codon that is as much as possible on the right of the gene, almost towards the end of the gene. So if you have another isoform that ends here, we ignore that. We take this one here. Okay. Always the longest that we can do it. We clone this UTR, we use Gateway. I don't know if you are familiar with these technologies. It's a system to clone sequences that you don't know the sequences. So you can't use a restriction enzyme to cut and clone it. So this system is actually very useful because you add some chemicals that allow recombination between two vectors and swap your 3' UTR, whatever you clone out of it, into this expression vector. So it's very easy to work with. And we prepare our our approach here with the vector here contains this gateway element here and here, so you can swap every 3' UTR very easily within these vectors. And so we decided initially, I told you 2,000, we're now shooting for 384, so doing 4 plates. We decided to focus on like a small data sets to optimizing the reactions. So we included transcription factors, genes involved in signaling UTRs of genes involved in signaling pathways, genes involved in cancer was pretty known to be involved in cancer. We took genes that are known to be targeted by LED7 and miR10B because we want to use this as a C2 microRNA to screen. And also we want to look, we included also all the bioinformatically predicted targets for the specific genes because also we want to see how sensitive is this approach here. And we spent an enormous amount of time optimizing conditions for melting temperature and PCR condition to clone this UTR. So these are the first, this really came up like last week. So these are 2 plates, these are all the UTRs that we start cloning, and now we are cloned and moved into this expression vector. And now Justin is just testing them into the screen. So unfortunately, again, I can't give you the finalized results on that. So, but the problems that we have with this approach is that you're going to just have a yes or no answer. If the microRNA recognizes the 3' UTR, you don't detect light, but you don't know exactly where the microRNA bind. And I told you that we really want to map the microRNA target. Okay, so how to escape this and the way we overcome this problem is that we add these repeats here, what they call MS2 repeats. Those are repeats that come from a bacteriophage. And these repeats are really important because they form these loops here in the cell, and they're recognized by a protein that is called MS2 binding protein that binds these repeats. So we took advantage of this system. We engineerize cells to express this MS2 binding protein. So when you express your construct into the cell express the protein is going to recognize these sites here and going to bind here. Okay. So it's going to bind here, so we can do an IP and remove out all the proteins that are exogenous, the one we transfect, so we don't have any more endogenous messenger RNA. Then what we're going to do a second IP using the RISC complex, and we're going to precisely map, degrade the RNA and perform kit clips and precisely map the sequence where the microRNA target within the 3' UTR. And the idea is a big picture that we want to really study how these networks, how the network look like, and how they change in disease. What is the dynamics of this network? You may have a microRNA that recognizes 100 genes, and you won't ever express it all the time. But then if you want to have that specific gene under the regulation, you may have a long UTR, so the gene is repressed in that way. Or sometimes you have a short UTR if you want to escape, right? But we want to really understand what are the rules behind this. It's not just pairing, base pairing. We want to study the biology behind that more than just computationally predicted. So we have a system. Now, I didn't put it in here because it's another big story, but we are collaborating with Karen Anderson in the biodesign, and she has very nice systems called the Georgetown method, which is a system to basically grow in plates primary tumors. And the way you do this, you basically use feeder cells that release a specific substance that allow the tumors to grow and thrive into these plates. And then you can recover these cells. And what she's interested in studying how this cell changes between, because what I told you, you can do this pair. You can have match pair. You can have normal tissues versus cancer tissue. So you can grow this in this system, then you can recover these cells and perform transcriptome sequencing runs. That's what she's doing to detect what are the factors that changes the protein, the changes in the network of gene expression that regulate and allow these cells to become more metastatic or less metastatic or not. So we are interested in is she's going to give us a few samples, and we're going to extract microRNA population. We're going to study the microRNA population of these cells, and since we can perform this experiment and sort of test whatever how this microRNA network change based on the stage of a tumor versus another, or subtype of tumor versus another. But this is a long-term shot. It's going to be really complex to do. So. But what we hope that this system will allow us to develop a system that we can use to sort of not just predict, because at this point we don't predict. We have a goal set of validated microRNA targets. So it's a lot more powerful than using prediction to detect these microRNA targets. So in conclusion, I told you that 3' UTR, I hope you go back to the lab, you're going to start thinking about your preferred gene and think about, maybe I haven't really thought about the 3' UTR. Maybe the reason why the protein is not expressed, or maybe I have a different isoforms. There may be more regulation than what I think to the specific message. Okay? It's involved, they really play key roles in many biological processes. They have this element that regulate the expression of the messenger RNA. Again, many people do sequencing run. They find RNA. They think that RNA is expressed, and they think the gene is overexpressed or downregulated. But at the end of the day, it's very difficult to study how the post-transcriptional element, the contribution of post-transcriptional regulation to a given to a gene, to the expression of a gene. Those are very difficult experiments to do. You need to incorporate both the transcriptome, the proteome, to sort of understanding the fact that I don't see a gene there as an RNA. It doesn't mean that I see a gene there as an RNA. It doesn't mean that eventually the gene is going to become a protein. Is the point. So they can induce profound changes in gene expression. It turns out that every organism possesses these multiple 3' UTR isoforms. We don't really understand how they are processed and what mechanism regulate this. But what is key is the dysfunction in this 3' UTR processing seems to be associated with many diseases. And I just mentioned about cancer, but it's also true in Alzheimer, in autism, and I can go forever. If you take your preferred disease and you start searching for it, I guarantee you will find a disorder within the 3' UTR of a microRNA that somehow is regulating or overexpressed in that disease and so on. So I'm not saying that this is the reason why the disease is caused, but definitely is one part of the disease that is poorly understood at this point and require more work for that. So these are the people that in my lab. There's Justin, Alex, and Steven. Then I have an enormous amount of undergrads that do a fantastic job in the lab. And this is me, and I'm located in the biodesign, and I stop here and I take questions. Thank you very much.
So we're a little long, but
sorry
for one question. Questions?
No questions. Good.
You worked with some of our students, right? Emmanuel.
Yes. Yes. He's great. Yes.
A few others, I think, as well.
Yes.
Or sort of collaborating with
We didn't
directors?
overlap. We didn't overlap with this project. Emmanuel is more interested into studying the contribution of human papillomavirus to the disease, to cancer. There are a few strains of human papillomavirus that are very highly tumorigenic, and he's trying to study them. But yeah, we didn't collaborate precisely on that.
Can you talk about your contribution to the Mod and Code project?
Okay, so do you know about modern code? Okay, so
Well, I know about ENCODE.
okay, so ENCODE turns out to be extremely complicated. So they decided to look into these ENCODE regions, like 1% of the human genome. Turns out to be extremely complicated also there to look for elements. So they switched on model organisms. They asked help to people who work on model organisms to look for elements. So I was in Fabio Piano's lab, and I was working on 3' UTR. So within this modern code, both in fly and human, people look for whatever element you can think of, right? Insulators, promoters, exons, and so on. So we were focusing on 3' UTR. So I was in Fabio Piano's lab for this project, and we were interested in really mapping the 3' UTR, where the boundary, where it starts and where it ends. What I was in charge of, I designed the pipeline and I physically cloned every 3' UTR, at least one 3' UTR isoform out of worms. So I spent all my postdoc basically with 96 well plates and tethered PCR machines and performing 3' rays like every day. I did like I don't know how many thousands and thousands PCRs. And I move all these UTR in vectors. I sequence them. I have the library here at the ASU, and I'm using this library to study the 3' UTR. Unfortunately, I didn't have much time to go into that part, but the first topic when I mentioned about the work that Stephen is doing with worm, looking for these tissue-specific RNA libraries, is to sort of like associate this kind of elements, kind of sequences with the 3' UTR that we have cloned. Because ideally, I want to look for what are these elements within the UTR that are important. And worms here is more powerful than human, because we know a lot more in worms than in human about this. Yeah, but basically I work cloning these 3' UTR, mapping them, sequencing them, and I have expertise both on the bioinformatics side and in the wet bench part. And that's it. I don't know if you need more.